All of our dreams can come true if we have the courage to pursue them. These words are not just mere words of existence, but rather my pathway towards the pursuit of excellence. I, Rudransh, a student researcher, do hereby present my research paper with you all, which is the cure to cancer. Now, before I begin explaining you the complex terms and definitions of biology, I would like to give you an overview about how we encounter the cancer disease. Cancer cells are already present inside our body, and under some favorable conditions, they start dividing, which leads to we people encountering the damn thing, which is the cancer disease. Now, the known cures to human beings are chemotherapy and immunotherapy. Now, chemotherapy, being the most popular one, kills cancer cells. Though it kills cancer cells, but it kills healthy cells as well. And I believe, what's the point of curing cancer if the person's gonna die anyway, right? Secondly, we have immunotherapy. It is promising, but yeah, it will cost you thousands and thousands of money. It's just like very expensive. So I kept these things in mind and I began researching on cancer and the possible cancer cures. And then finally, I came up with OncoSyn-x. So OncoSyn-x is like a virus which is programmed to find cancer cells. It's just like a police officer searching for thieves. So this virus goes inside your body and searches for cancer cells. And there will be a point when cancer cells will divide, right? It will find that area and it will stick that cluster of cancer cells which is in the making. And then it will burst itself, meaning that it will initiate its cell apoptosis or programmed cell death. Just be familiar with the word because I'll be using it way more frequently further. Now, over the next few minutes, I'll be representing my screen and I'll explain each and every term that I have mentioned in the paper so as to make it easier for you guys to understand what it really is. Right, this is my research paper. Abstract background methods, results. So here I've just, for the sake of the formatting of research paper, I have added these proper headings and the words here, sentences. So these specifically contain what cancer is and what is OncoSyn-x. If something is very useful here, you can read this paragraph. A fully synthetic oncolytic virus designed from modular genetic elements. Here I am just talking about some basic science of, or basically my anatomy of this virus, and of course I'll share with you as we go on and on this research. Introduction. Almost the same thing I explained to you about cancer earlier, so I've written the same thing here, just with more credibility of internet as well. Alright, so now we have materials and methods. What is synthetic gases design? So I'm just discussing about the morphology of the virus. It contains ligands, and these ligands are programmable to specific promoters found in cancer cells. Usually they are proteins such as the HER2, as I've mentioned here. Yes, I've mentioned HER2, EGFR, and CEA. CEA is not so much of it's not very much useful, but these HER2 and EGFR are very useful. HER2 stands for human epidermal receptor 2, while the EGFR, it stands for epidermal growth factor receptor. So these two are found in cancer cells, and these receptors help my protein to detect cancer,
and then to stick itself. These ligands, they stick to the cancer cell. Alright, now we have the promoter selection for tumor specific replication. Now, the main thing, to ensure specificity of cancer cell, my virus contains specific tumor specific promoters so that it detects it as well, and then after ensuring itself, it sticks to the damn thing, and then cell apoptosis happens. Programmed cell death. I know you now you'll be familiar with the word apoptosis. It just means cell death. So OncoSyn-x uses a dual promoter system to restrict viral replication to cancerous cells. Now, if I just break it down into very simple language for you guys, cancer contains two specific promoters which promotes cell division, and secondly, it prevents cell death. The first one is the HTRT promoter. This HTRT promoter promotes cell division, cell replication, which helps cancerous cells to divide themselves and make, a crowd of cancer cells, which is harmful for the body, of course. And the second one is survivin. This survivin promoter just prevents cell death. So I've written here selectively active in apoptosis resistant tumors. So it just prevents cell death, apoptosis resistant. Now, talking about therapeutic payload encoding, I've used three major proteins here: trail, holin and endosilin, and thirdly the GFP protein, or GFP, green fluorescent proteins. First one is trail. This trail helps to do the apoptosis. So the whole apoptotic process is executed by trail here. Now, if I talk about holin and endolysin, this endolysin and holin, their work is limited to cell lysis. So they just open the pathway for the trail to do its work, to go to the next stage of cell death. Talking about GFP, this GFP helps in tracking of the virus. So GFP itself means, GFP means green fluorescent proteins. So if I were to look into the microscope to check if the virus is working correctly or not, then if I use the GFP here, I'll see a green slimy substance moving here and there and doing its work and detecting the cancer cell and then sticking to it and then going inside it. Soon I'll be making an AI simulation of this thing, and it would be better for you. You'll understand it better. So this GFP is only limited to tracking of this particular work. Now, about kill switch mechanism. A TETR based kill switch is inserted downstream. These are just the formal languages I've used here to be specific, or to break it down for you guys to understand it. The kill switch mechanism is that there's a what if that arose in my mind when I was writing this paper, that what if this virus gets mutated at some other place in our body, and by coincidence it detects the same promoter in some at some healthy site. So what if this virus finds a promoter inside the body, and hence it would go and, as we all know, it would execute the apoptosis. But this scenario is really, really rare, because my virus is very specific when it comes to killing these cancer cells. But still, if I take it as a possibility, I've developed a kill switch. So if I just inject this drug inside the body while the virus is working, it will activate the synthetic gene, the synthetic suicide gene, which is present inside the virus as well, which is inducible DNAs, and it will directly kill itself, halting replication and triggering genome degradation. Right, now I've just So I had used this simulation, and this was the simulation, but it was good. This is the diagram I had developed, I had made. So this is the Caspian engineering. This is the kill switch. Kill switch apoptosis lysis inducing genes and cancer cells. That's yeah. So that's how this. So this diagram shows the schematic structure of OncoSyn-x virus and the gene control logic, of course. Now the discussion part. The design of OncoSyn-x highlights the potential of. So this discussion part just is again, I told you, I've I had to write this for the sake of the format of research paper. It doesn't consist much informational part of the whole research. Concluding the thing and now the future work. Hence, OncoSyn-x can be used in various things. It can be induced into bacterias, and it can be, we can use non-viral bacterial and yeast systems, and since it contains logic of promoter-driven replication, kill switch and all, it might help further. And but my future goal relating to this research is to build an AI simulation and to present it at various scientific conferences, and to bring a revolution in the field of biology and the treatment of diseases, such detrimental diseases like and cancer, Alzheimer's, Parkinson's, and as well as the diseases as well. I haven't researched about heart diseases, but I'm planning to research on it, and I'll do it later. But this is my project about oncolytic virus, in which is a programmable gene circuit. All right. Abbreviations. These are all the things I used. These are all the definitions. So yeah. So references I've used it from the research paper of 5 people. First one is launched in 2012 and all. So my work. You might hear the word oncolytic virus later on, and you might think that this guy is just copying down the thing. The difference between oncolytic virus and OncoSyn-x virus is that oncolytic virus is developed from natural viruses. It is like the natural viruses, they are modified and then they are converted into oncolytic viruses. So, firstly, they are not that much specific, plus, there are just 50 to 60% chance that it would encounter the correct place that it will work on. So it's like no surety can be given to the natural oncolytic virus. But when it comes to OncoSyn-x virus, this virus itself, it is designed from scratch by me myself, and of course this whole thing I just designed. So basically that's it. That's OncoSyn-x, and I hope you liked it, and by now you might have understood everything related to this research. If you guys have any queries related to this research paper and the information I shared with you, feel free to ask in the comments, and I'll be very happy to respond to answer your queries. Thank you.